Hello dear students, I welcome you all for our lecture series on agrochemicals and pest control. The topic for today's discussion is biotechnology approaches in pest management. Plant biotechnology plays a key role in the massive production of improved crop varieties as well as in their genetic improvement. They can also help in propagating plant species which contain useful and biologically active substances, example food additive, pigment, pharmaceuticals and biopesticides. Organ, tissue and cell culture could be more efficient than conventional extraction. Due to increasing problems occurring from massive applications of pesticides such as insect resistance to pesticides, the use of biotechnological tools to minimize losses from insect pest has become inevitable. Presenting alternative strategies for elevating biotic stresses, biotechnological approaches for pest management and ecological sustainability explores how the modern tools of biotechnology can be used in pest management for sustainable crop production, the biosafety of transgenic crops and environmental conservation. Now, in this episode, the following aspects are going to be seen: seed technology, herbicide resistance, insect resistance, virus resistance and application of plant tissue culture to agriculture and forestry. Let me start with our first objective, seed technology. Seed technology is the methods through which the genetic and physical characteristics of seeds could be improved. It involves such activities as variety development, evaluation and release, seed production, processing, storage and certification. Seed technology includes the development of superior crop plant varieties, their evaluation and release, seed production, processing, seed storage, seed testing, seed quality control, seed certification, seed marketing, distribution and research on seed, these aspects. Seed production, seed handling based on modern botanical and agricultural sciences. Coming to scope, India is a vast country and bestowed with varied soils and has got different agroclimatic zones enabling year-round cultivation of crops. By and large, most seed crops are grown during Kharif season. However, most of the vegetable crops are produced in Rabi season and they possess better quality seeds than the crop grown in Kharif. Indian farmers can practice with multiple cropping system. The farmers can opt for different crops like cereals, pulses, oil seeds, vegetables, etc. in all the three seasons viz. Kharif, Rabi and summer. With the advancement of agriculture, the government of India felt that there is a need to establish seed technology department in Agricultural University and Indian Council of Agricultural Research Institutes in India after the recommendations and suggestions given by National Commission on Agriculture. Accordingly, the seed technology department was initiated throughout the country with the following main objectives. They are to teach seed technology course, research on seed production, processing and testing, to strengthen the seed technology research, to give training to those who are involved in seed production, processing and testing. Goals of seed technology are rapid multiplication. that is to increase agricultural production, and timely supply, so that new varieties must be available in time. Next is assured high quality of seeds with good vigor and viability. Fourth one is reasonable price, that is cost of seed must be low to reach the average farmers. Moving on to herbicide resistance. Genetically engineered herbicide tolerant crops are likely to be the first commercially available products. They are deeply embedded in the chemical quick mix mentality. Herbicide tolerant crops are engineered to contain new genes that help plants avoid the harmful effects of particular weed killers. Currently, a crop sensitivity to a weed killer limits the amount of herbicide growers can apply. With herbicide tolerant crops, farmers can be persuaded to use more of a particular herbicide to kill weeds without damaging their crop. Herbicide tolerant crops represent a simple strategy for chemical companies to market more of their herbicides. All 8 major transnational pesticide companies are currently funding research to develop a variety of crops that tolerate their herbicides. Monsanto, for example, has already field tested genetically engineered glyphosate tolerant tomato, cotton, soybean, flax and canola. Studies link various weed killers with cancer, nervous disorders, behavioral changes and skin disease in humans and animals. In addition to poisoning farm workers who handle herbicides, weed killers enter groundwater and other drinking water supplies, contaminate food and destroy wildlife and their habitats. Furthermore, the transfer of genes for herbicide resistance to weedly relatives could make some weeds more difficult to control in agricultural settings. It is virtually certain that herbicide tolerance genes would be transferred via cross-pollination from the engineered crucifers to wild weedly relatives, resulting in weeds resistant to herbicides and therefore more difficult to control. Next is insect resistance. Reducing crop loss caused by insects is also a major focus of agriculture biotechnology research. Monsanto, Ron and Haas, Agrigenetics, Advanced Sciences, Calgene, the USDA and the University of California have developed and field tested tomato, tobacco, cotton, walnut and potato plants genetically engineered to contain an insect killing toxin from Bacillus thuringiensis. Sandoz Crop Protection and Crop Genetics International are genetically engineering microorganisms containing Bacillus thuringiensis toxin to act as biocontrol agents. Bacillus thuringiensis is a soil microorganism that has been used for 20 years as a commercial biocontrol agent against certain insect pests. Knowing that specific toxins were responsible for Bacillus thuringiensis insecticidal activity, genetic engineers have isolated and removed the genes that produce the toxins and placed them in plants and microorganisms. Engineers are designing Bacillus thuringiensis containing crops, trees and microbes to combat an array of insect pests. European corn borer, cotton bollworm, Colorado potato beetle, beet armyworm, tobacco hornworm and tomato fruitworm. Despite their promise for reducing the use of chemical insecticide, widespread use of Bacillus thuringiensis containing crops and microbes poses a potentially significant problems. Accelerated evolution of pest resistance to Bacillus thuringiensis. If this were to happen, agriculture would lose one of its safest, most valuable biocontrol agents. Already some insect populations, example diamondback moth, have become resistant to the Bacillus thuringiensis toxin after prolonged exposure. Resistance in a particular insect pest population means that Bacillus thuringiensis would no longer be effective in controlling that pest. It is generally accepted that the intensive use of Bacillus thuringiensis in genetically engineered organisms will accelerate the selection pressure on insect populations to develop resistance. In engineered plants that produce the Bacillus thuringiensis toxin throughout the life of the plant, insects will be exposed more frequently and for longer periods. This intensified selection pressure contrasts with conventional methods of delivering Bacillus thuringiensis where the toxin is active for only a limited period after application. Moving on to the next component, virus resistance. Viruses cause economically important diseases in most of the major agricultural crops. Thus far, there are no chemical viricides that do not also harm crops. Some crops are treated with insecticides to kill insects that carry viruses from plant to plant. Plant genetic engineers have suggested a new approach to controlling viruses. They are engineering plants to contain a virus gene. The plant then produces a viral protein which enables the plant to resist attack by the same virus. The result is similar to immunities created by vaccinating people and animals against diseases. Monsanto, Cornell University and the University of Kentucky have field tested genetically engineered virus resistant plants including potato, tomato, tobacco, alfalfa, cucumber, cantaloupe and squash. Adoption of virus resistant plants may in the short term reduce the use of chemical insecticide and losses due to viruses. For the long term, however, it remains unclear how fast viruses might evolve resistance to virus genes incorporated into resistant plants, rendering those engineered plants once again susceptible to virus attack. Crops genetically engineered to resist herbicides, insects and virus diseases like chemical pesticide will be sold to farmers as single, simple to use products to control pests and sustain continuous monoculture. They are being developed to fit immediately and easily into conventional agriculture's industrialized monocultures, and as such they extend what Jack Dole has called the invade and conquer and replacement parts approach to pest management. This kind of farming entrenches farmers' dependence on successive new products from corporations, new genetically manipulated organisms to serve as quick fixes for increasingly complex pest control problems. Now coming to application of plant tissue culture to agriculture and forestry. The most important challenge in a developing country is food for its growing population. The arable land area is fixed. Some means has to be there to increase the productivity. Plant tissue culture technique offers an excellent opportunity for mass propagation of plants in laboratory test tubes, which are transferred to the field. Besides croplands, the technique is also applied to regenerate saplings for plantation and regeneration of diminished forests. Some rare and nearly extinct plant species can be rescued and propagated by this technique. Embryos produced by incompatible crosses also are rescued. Seed dormancy is overcome, life cycle is shortened and much more. This technique is merged with genetic engineering to regenerate plants with novel characters and combine two or more beneficial characters into a single plant. Things once seemed impossible have been made possible. In this section, we shall discuss about 4 elementary applications of this technique, namely micropropagation, organogenesis, somatic embryogenesis and protoplast culture and fusion. First one is micropropagation. Basically, micropropagation is similar to rooting of plant cuttings and is in a way another method of vegetative propagation of plants. However, it differs from the conventional procedure in that it is carried out in an aseptic condition and requires a unique recipe, that is an artificial nutrient medium. It is used for forestry improvement and is an example of direct laboratory to land transfer of biotechnological benefits. A small plant cutting or explant is surface sterilized and inoculated into a culture vessel containing a semi-solid nutrient medium. The inoculated culture vessel is incubated at room temperature. In a day or two, a large number of shoots develop from the axillary bud in a process known as axillary bud proliferation. Each growing point is subcultured to give rise to shoot. This phenomenon is known as adventitious shoot formation. Each shoot is stimulated by an auxin to develop roots. The new plantlet is transferred to the field. Next is somatic embryogenesis. In flowering plants, an embryo is the product of the zygote and the zygote is the product of the fusion of two gametes. The embryo undergoes a pre-programmed development and forms a plantlet. In a nutshell, under normal circumstances, an embryo is the result of sexual reproduction. Such embryos are known as zygote embryos. However, plant tissue culture technique offers a method of producing embryos from somatic cells bypassing sexual reproduction, such embryos known as somatic embryos. The process of formation of somatic embryos is known as somatic embryogenesis. Somatic embryo formation starts with a mass of single cells or a tissue grown on a semi-solid nutrient medium. A cell repeatedly divides and forms a cell aggregate. The cell aggregate passes through different stages such as globular, heart-shaped and torpedo-shaped stages. The torpedo-shaped stage is the mature stage. The culture is initially started on a semi-solid medium and the callus so formed is transferred to a liquid medium in an agitated and aerated bioreactor. The cells breaking off from the callus develop into somatic embryos. Mature stages are sorted out and grown to maturity on a semi-solid medium. Dormancy is induced before these are processed for transfer to the field. There are 4 methods by which the somatic embryos are transferred to the field. First one, these are germinated in the laboratory, transplanted in pots and then transferred to the field. Or the dormant embryos are encapsulated in a gel containing an adequate nutrient for the embryo. These encapsulated embryos are known as artificial or synthetic seeds. Such seeds can be planted in the field. Or this method involves the germination of the embryos under a controlled condition and then mixing of the seedlings is mixed with a gel-like medium. The seedling gel mix is sown in the field. Fourth one is the embryos are germinated and then fluid drilled. The major advantages of this process are rescue of zygotic embryos formed by incompatible crosses and overcoming seed sterility and dormancy. Coming to protoplast culture and fusion. Protoplasts are plant cells whose cell wall is digested. The cell is bound by a plasma membrane. Isolated protoplasts from 2 different species of plants have been successfully fused to produce a single protoplast containing the genetic material and the cytoplasm of both the fusing protoplasts. The process of fusion is not straightforward. It is facilitated by some agents known as fusogens. Fusogens are of 2 types, chemical and electrical. Polyethylene glycol is a chemical fusogen. It is not used as a universal fusogen since it is toxic to protoplasts of some plants. Alternatively, pulses of electric current are applied to the fusing protoplasts. This method is known as electrofusion. This depicts the sequential fusion of 2 protoplasts resulting in a synkaryon. There are 3 steps in this process. First, 2 protoplasts come in close proximity, then the plasma membranes of the 2 fuse and then the 2 nuclei lie in the mingled cytoplasm. This stage is known as heterokaryon. In the 3rd stage, the 2 nuclei fuse forming a synkaryon. The cell wall of the fused protoplast is regenerated and the cell is cultured in an artificially enriched nutrient medium. The following sequence of events is same as that in the callus culture. This process is also known as somatic hybridization and the products as somatic hybrids. This method bypasses the fusion of gametes of 2 unrelated plant species. P. S. Carlson, H. H. Smith and R. D. Daring obtained the first somatic hybrid by fusing the isolated protoplasts of Nicotiana glauca with Nicotiana longsdorffi. However, sometimes due to cellular incompatibility, 2 nuclei cannot coexist. Consequently, one nucleus is eliminated and a protoplast containing the nucleus of one species and the cytoplasm of both results. The ensuing hybrid is known as a cytoplasmic hybrid or cybrid. Somatic hybridization is attempted in plant species which are sexually incompatible. The well-known example of a somatic hybrid is pomato, obtained from the protoplast of potato and tomato. However, this hybrid is of little commercial value. Before I finish, let me conclude this session. To conclude, I can say that biotechnology is frequently deliberated the similar with the biomedical investigate, but there are a group of other industries which take advantage of biotech method for studying, cloning and varying genes. We have turned out to be familiar to the thoughts of enzymes in our everyday lives, and a lot of people are recognizable with the argument adjacent the use of genetically modified organisms in our foods. So students, hope you enjoyed this session. Thank you so much for watching this episode. Keep watching our programs. We will meet in the next episode. Till then, take care.